so subdued this morning? Friday. Questions? Quiz, remember, on Monday is section 13.4. Kind of the DNA technologies that overlap the class. Other than reading, anything else to do, Dr. Morgan, for preparation for Monday?
Read the section under week 2.
Read section under week 2 in your lab. Okay, we began talking about the operon last time, and remember, an operon is a set of genes which allow bacterial cells to regulate gene expression depending upon what's happening in the environment of those cells. We talked about some elements that make up the operon. Anybody know what any of those elements are? What are the parts of an operon? There's a region called the operator. And the operator is actually a part of another region that we've talked about previously that really is important for all genes in terms of transcription. What region is the operator part of? It's part of the promoter. And what molecule has to be able to dock onto that promoter and then move down it and transcribe those genes? RNA polymerase. Okay? So the operator is one really important element of the operon, and whether this operator is blocked or not really determines whether transcription can occur. Okay? What can interact with the operator and block it? There's a protein called a repressor protein. That can interact with the operator. Those are those orange trachycones, if you like, repressor proteins. And if they are bound to the operator and the operator is blocked, transcription doesn't occur. If they don't bind, operator is open, transcription can occur. So remember that these repressor proteins exist in two forms, or two states, two conformations. An active state and an inactive state. When they're active, they bind to the operator. When they're inactive, they don't. What's going to determine whether a repressor is active or inactive? Its shape, and what's going to help determine its shape? No? What's binding to it, okay? Because these are proteins, and other molecules can bind to proteins and change their shape. Okay? And there's a gene, the repressor gene, that codes for this repressor protein. This particular gene is always active, so we're always producing this protein. Okay? What's the third element of the operon? We've got repressor genes. We've got operators that are part of a promoter region of the DNA. What's the third element? Structural genes. And remember, structural genes, plural, usually there are a bank of genes that are all kind of regulated together. For example, in the trip operon, to manufacture tryptophan, there are 5 amino acids that you need to, or 5 enzymes you need to produce in that biosynthetic sequence of making tryptophan. If we're going to make tryptophan, we need all 5 of those. If we're not going to make tryptophan, we don't need any of them. And so these are all regulated kind of as a block, and they're turned on or turned off together. Okay, we're going to use 2 examples of kind of contrasting types of operon. The lac operon and the trip operon. Which one is involved in regulation of catabolic metabolism, breakdown reactions? Black. Trip, anabolic metabolism. So here in this particular case, synthesis of an amino acid, tryptophan. Okay? Degradation in this particular case of the sugar lactose into, what are the two monosaccharides that make up lactose? Lactose is a disaccharide. Glucose and galactose, which are both 6-carbon sugars. They're isomers of one another. Anybody know the name of the enzyme that can hydrolyze lactose down into glucose and galactose? Lactase, or more appropriately, beta-galactosidase. So that's the enzyme that can catalyze this reaction. So this is one of the things we're going to be regulating in the lac operon, is this beta-galactosidase enzyme. Okay, so let's take a look at the trip operon first. What's the normal kind of default situation for the trip operon? An unblocked operator. That's the normal default situation. So if the operator's unblocked, are those structural genes to make those 5 enzymes for tryptophan synthesis going to be transcribed? Operator unblocked. Transcribed, yes or no? Yes. Okay. Those enzymes are going to be transcribed into messages. Messages will be translated into proteins, enzymes, and the biosynthetic pathway will work. Okay? Under what conditions will we want this to be turned off? There's already a lot of tryptophan there. Okay? Where do a lot of bacteria live? In your gut. Okay? You've got gut bacteria, including E. coli, which have both lac and trip operons. Do E. coli need tryptophan to grow? You bet. You bet. And if they can't get it from the environment, then they will synthesize its own tryptophan. Well, what if you have a big turkey dinner? Turkey meat is relatively high in amino acid tryptophan. You digest your turkey. You break it down into amino acids. Those amino acids are transported across the intestine into the blood, and your cells get some tryptophan, but so do some of the bacteria that are living in your gut. So they're kind of bathed in tryptophan. So, keep the trip operon on then? No, let's shut it off. Let's not waste energy making those enzymes. So what's going to turn it off? High levels of tryptophan is going to turn it off, right? But how does that flip the switch? Mechanistically, what's going to turn off this operon? What's going to actually physically happen in order for this operon to be turned off? The repressor protein is all of a sudden going to start binding to that operator. Well, what's going to cause it to do that? Tryptophan is going to bind to the repressor protein and change its shape from the inactive state to the active state. And now it can bind to the operator, and now it shuts off that particular transcription process. I think it's really important for studying for these kind of molecular things to pay close attention to diagrams. And for the next exam that's coming up, which will include lac and chip operon, there are really good diagrams in your textbook, and they're the same diagrams I use here. So if you can look and kind of reason your way through a diagram like this, then you know what's going on. Could you explain this diagram to someone else? Could you sit here and say, okay, here's what's going on? So, here is our trip operon, consisting of a promoter, 5 structural genes, and then here is this regulatory or repressor gene. That makes a repressor protein. But as this is synthesized, the normal state for this particular protein, the normal shape, is an inactive shape. What does that mean? It means it can't bind to the repressor, the operator, sorry. So this doesn't fit here. So RNA polymerase settles in and recognizes this promoter region. Operator isn't blocked, and so it just slides on down and begins to transcribe those genes. And then those are translated into proteins. And those are the enzymes that we need for the biosynthesis of tryptophan. So this is the normal status, operator unblocked, operon active. We make the enzymes. Now, if there's a lot of tryptophan present, that can interact with this repressor protein. And when it binds to the repressor protein, it causes its shape to change. Now this repressor is in the active state. So what does it do? It can now bind to this operator. If the operator is blocked, RNA polymerase can't get past those traffic cones, and transcription is stopped. We don't make the enzymes. We don't synthesize our own tryptophan because we've already got it present from the environment. And as long as there is enough tryptophan present to keep interacting with repressors and keep blocking this operator, that operon will be shut off. But what happens after you digest your turkey material, your turkey dinner, and all of a sudden you decide, I'm going to go on a meat-free diet for a while. And I'm going to start eating other things. Well, now you're not digesting protein. And now the amino acid levels in your gut are going to start to drop, including tryptophan. And eventually this will be used up to make protein. So now there's not enough to interact with this repressor protein. So the shape of the protein switches back to the inactive state again. Can't bind to the operator any longer, and transcription will start again. Okay? So the molecular signal is an environmental substance which also happens to be the end product of a metabolic pathway, the amino acid tryptophan. See how important it is that proteins can interact with stretches of nucleotides on the DNA? A protein-DNA interaction is going to be something that's going to be responsible for regulating transcription. Okay, let's take a look at the lac operon. It's pretty much just kind of the opposite of the trip operon. What's the normal status of the lac operon? Off. We don't transcribe those genes. Under what conditions do we want to transcribe those genes? if the substrate that those enzymes break down is present. So the signal for turning on will be the presence of lactose. So again, let's go back to your gut bacteria. If you're not consuming a lot of lactose, maybe you don't like milk, maybe you don't like dairy products, then bacteria will preferentially use other things in your gut, like glucose, to support their metabolism. However, if lactose becomes available, under certain conditions we'll talk about in a minute, they'll start using that lactose. Well, there are 3 structural genes that are involved in lactose metabolism. What's one of them? The one that codes for this enzyme, beta-galactosidase. So that's one structure. There's a second structural gene involved in lactose metabolism called the permease gene. And this actually codes for an enzyme, or a membrane transport protein, a carrier molecule, which helps to get lactose into the cell. It's hard to metabolize it if you can't get it into the cell. And then there's a third protein called transacetylase. And we're not absolutely sure what role that plays in lactose metabolism, but we know it's required. So again, if lactose isn't present, then do we need the beta-galactosidase enzyme? No. Do we need this? No. Do we need this? No. So let's just not produce any of these enzymes. Let's shut off the genes that code for these enzymes. But if lactose becomes available, let's switch this system on so we make all these proteins that are needed for lactose metabolism. Okay. So here's the lac operon. Here's our regulatory gene that codes for the repressor protein. Here's our operator. Here's our promoter. And then down here, there are some of these structural genes. In this particular situation, the repressor is in the active state as it's made. When it's translated into a protein, the shape of that protein is correct to fit the operator. Operator blocked, polymerase can't move past it, no transcription. So the normal status is off. What's going to turn it on? Presence of lactose. How's it going to do that? Lactose is going to interact with this repressor protein and change its shape to what? Inactive shape. So now it can no longer bind to the operator. Transcription occurs. Okay? So, in the presence of lactose, here's what's happening. RNA polymerase is transcribing these 3 genes, galactosidase, permease, transacetylase. Those are being made into proteins. And then these will catalyze the conversion of lactose into the 2 monosaccharides. These can now be used in cellular respiration to do energy. This cannot be used directly in cell respiration. This has got to be converted into these for this sugar to be an energy source. So in the presence of lactose, and actually it's a metabolic product. It's a kind of a slightly converted form of lactose called allolactose. Don't worry about that. In this particular case, lactose is said to be the inducer. And its presence activates this operand. Why? By inactivating the repressor protein. So the operator is no longer blocked. Protein gene or protein DNA interactions, again, regulating gene expression. Now, both of these are examples, the lac and the tripoperon, of what we call negative control. In negative control, environmental factors, either tryptophan or lactose, turn on or off the operand. So negative control kind of is an on-off switch. There is also genetic regulation that involves positive control. And this is more of kind of a volume control. Okay, if you think of a stereo receiver, you've got to turn it on or off to get your receiver working. If it's turned off, no output. If it's turned on, there's output. But you also have a volume control, too, that says how loud the music can be. In this particular case, the volume control is the rate of transcription. Remember, just because something is on doesn't mean it is fast. So the lac operand has both of these positive and negative control mechanisms working.
Why are they called positive and negative?
Don't know. I mean, it's a good question. And actually, your textbook attempts to answer that question, and I found their explanation to be kind of incomprehensible, to be honest with you. So for my purposes, if you think of just negative control, meaning on-off, positive control, meaning volume, that's conceptually what I want you to understand. Okay. Sometimes the derivation of terms is a little bizarre, and it has to do with kind of research outcomes. But I don't know, Larry, can you give any more insight into
I
that?
always found this to muddy the water.
Yeah,
So it's just something you memorize.
yeah. Sorry about that. Okay, so the lac operand has both these positive and negative control elements. We've already talked about the negative control elements, the on-off switch. Okay? Let's talk for just a minute about this positive control that can occur. Okay, E. coli bacteria are a little picky in the kind of foods that they like to use. They prefer glucose. And if both glucose and lactose are present, they'll preferentially use glucose. Okay? If lactose is present and no glucose is present, then they'll use the lactose. Okay, that's important in understanding this positive control. Another part of this picture. Cyclic AMP. You've heard of cyclic AMP before. Where does cyclic AMP come from? ATP, okay. There are enzymes that convert ATP into cyclic AMP. And we've seen cyclic AMP in the context of being a second messenger in signal transduction pathways. You had a quiz on signal transduction. The levels of glucose and cyclic AMP are inversely related. And physiologically, we just don't have time to get into that relationship and why that's the case. But if you have high levels of glucose, your cyclic AMP levels are low. If you have low glucose levels, your cyclic AMP levels are high. So remember that inverse relationship. Okay, there's one more element in this positive regulation that we need to throw in here. And that's another protein called catabolite activator protein. This particular protein, if it's allowed to attach to the promoter, enhances the rate of transcription. So this is going to increase transcription rate. I don't know if you remember last exam. We said that RNA polymerase usually needs some help to position itself properly on this promoter and to transcribe the genes. We need some transcription factors. This is a transcription factor. And it really increases the rate at which transcription occurs. Now, it's got to be able to bind to this promoter. And in its normal state, it's inactive. But if there is cyclic AMP around, the cyclic AMP binds to the CAP protein, changes its shape to an active shape. That binds to the operator now. And then that enhances the rate of transcription. It goes faster. Well, when is this going to be high? When there's low glucose. If there's high glucose, remember, there won't be much of this. Therefore, this will stay inactive. And therefore, even if this operator is unblocked, this rate of transcription will be low. So this gives us our volume control. This cap protein can turn this from a really, really low rate of transcription into a really, really high rate of transcription. So look at this relationship again. The bacteria are preferentially going to use glucose. If both sugars are present, they're still going to preferentially use the glucose. Why? Because low levels of this will result in high levels of this, will result in activation, and we'll get a high rate of transcription. But remember that for this operator to be unblocked, lactose has to be present. So this interaction between the levels of the sugars and whether one sugar is absent or present will regulate the utilization of those particular energy sources. So this is a little bit more complicated. This is another wrinkle in here that gives us this volume control versus just the on-off switch. Now, what we're going to see in eukaryotic cells is that this is pretty simple regulation. Bacterial cells have pretty simple regulatory mechanisms for gene expression. Things get a lot more complicated in eukaryotic cells, and that's where we're going to go next, and we'll just barely scratch the surface on some of the opportunities there are to be able to regulate gene expression at the eukaryotic level as opposed to this more simplistic system of the prokaryotic level. Chew on this part for a while, and we will come back as the next, we're not going to go, as the next few class periods take place. And I'll come back and try to make sure that we review this lac trip operon, this positive and negative control, so you can wrap your head around it. Because this is not the kind of thing that just immediately usually clicks in people's minds and go, I've got it. It takes some work, okay? And so if you're confused right now, one of the things I would really like you to do over the weekend is look at these diagrams and go through them at your own pace and see if they can make sense to you. And if they can't, figure out where you're getting hung up, okay? What parts don't make sense to you and what parts don't. And then at the beginning of the period, when I ask are there questions, if there are places you're hung up, let me know. But this is the kind of thing we'll work on for additional class periods. Just come back and review it. Because this is kind of fundamental understanding of kind of a picture of how gene expression could be regulated in a simple prokaryotic cell. Regulation of gene expression in a eukaryotic cell is a whole other ballgame. Think about the complexities that eukaryotic cells have that prokaryotes don't. Number one, prokaryotic cells typically have a single chromosome. Our cells have 23 pairs of homologous chromosomes. In eukaryotes, proteins are associated with those chromosomes, and they can fold up those chromosomes into very highly compacted states. But what's really different about prokaryotic and eukaryotic gene regulation are some of the interactions that take place. And a big interaction that takes place in eukaryotes are RNA, DNA interactions. And that creates some really fascinating things that we've learned about the regulation of gene expression in eukaryotes. And this is a rapidly advancing field right now. We're discovering all kinds of new things about RNA molecules that are encoded into the DNA. And then when those RNA molecules are produced, how do they interact with the DNA to regulate gene expression? Now, remember that with the exception of your sex cells, all the cells in your body have the same genetic information. But those cells oftentimes are different cell types. That's the result of differential gene expression. Some genes being expressed in one cell, not in the other. What's interesting is that in any given cell, of those genes that that cell has, about 20,000 human genes, In any given cell, probably only about 20% of those are active in any given time. The remaining 80% are turned off. They might be turned off permanently, but more often than not, they're turned off temporarily. And under certain conditions, it might be environmental conditions that determine this. It might be a sequence in organismal development that determines this. Those genes could be activated and other ones turned off. The 20% that are turned on will differ from cell type to cell type. That's what makes the cells different from one another. Very importantly, if errors occur in gene expression and the wrong gene gets turned on in the wrong cell, or at the wrong time in the life of the cell, or if the wrong gene gets turned off, that can result in diseases, including cancer, or in the development of an organism, developmental defects. So researchers that are studying cancer and developmental biology are very much interested in genes and how they behave and how they're regulated and what turns them on and what turns them off, and how all of this happens in that precise fashion that results in a single-celled zygote developing into a multicellular organism with specialized tissues, and all those tissues work together to perform an intact functioning organism. And again, I've said it before and I'll say it again. It is totally amazing that all of this happens in a way that results in you as a zygote turning into you as an individual that is a healthy individual. Well, in eukaryotes, there are a lot of opportunities to regulate gene expression. In bacteria, gene expression is primarily regulated at the transcriptional level, okay, through things like operons. Turning on and turning off genes in response to environmental cues. Is tryptophan present? Is lactose present? Much more complicated, however, in eukaryotes. Okay, we've already talked about this a little bit, and there was one question on your exam last time about this. Let's go back for a minute to kind of the biggest picture when we talk about genes, and let's talk about chromosomes, and particularly let's talk about chromosomes during interphase of the cell cycle. Remember that during interphase, chromosomes are in this tangled state called chromatin, where all the chromosomes are kind of wound up and intertangled with one another. And one of the things that happens as we move through the cell cycle and get into the mitotic state is that during prophase, those chromosomes begin to condense, shorten and thicken, line up on spindle fibers, and then at metaphase, the duplicates are pulled apart so that each cell gets half the number of chromosomes. Okay. Well, chromatin structure is one opportunity that we have for regulating gene expression. We talked about the fact that the basic unit of chromosome is this structure called a nucleosome. I'll show you that in a second. And that proteins called histone proteins are responsible for kind of maintaining and forming this nucleosome structure. How the DNA is folded up into these nucleosomes can really help to determine whether those genes are available for transcription. So this is an opportunity for the cell to regulate gene expression by regulating how that DNA twists and folds up. Okay, so we've seen this before. So here's our DNA double helix. DNA is associated with, in eukaryotes, these histone proteins. And groups of these histone proteins can interact with the DNA. So the DNA wraps around the histone proteins. It coils. And so these are the nucleosomes. These are sometimes called beads on a string. Okay? But as a chromosome moves from chromatin into the more condensed state of a metaphase chromosome, these nucleosomes become increasingly tangled and twisted up into more and more compact structures. Oops. They loop around one another, so they finally make up this metaphase chromosome. Okay, here's something you should definitely keep in mind. The more tangled and compacted DNA becomes, the less available those genes are that are on the DNA are going to be for transcription. The transcription machinery just can't get to those genes when it's all packed up in this metaphase chromosome. These are pretty much transcriptionally inactive. The more we loosen the DNA structure, the more likely it is that RNA polymerase molecules can interact with the DNA and then transcribe it into RNA. So packaging and compacting DNA tends to shut down transcription. Loosely packed DNA is called euchromatin. Tightly packed DNA is called heterochromatin. Euchromatin is usually transcribed. Heterochromatin is not. Well, there are chemical modifications to the histone proteins and to DNA itself that can influence how tightly DNA is packaged up. So if modifications occur that encourage DNA to be in the compacted or heterochromatin state, that's going to tend to shut off gene expression. If modifications occur that result in DNA becoming less packed, euchromatin, then that tends to favor transcription. These two modifications include acetylation and methylation. There are enzymes that can add acetyl groups. There are enzymes that can add methyl groups to histone molecules. And when you start adding molecules to histones, which are proteins, their shape changes. And if histone shape changes, that's going to influence how DNA wraps up and coils up. That's going to influence whether DNA is euchromatin or heterochromatin. There are also enzymes that can actually chemically modify DNA itself, not the histones now, but the DNA molecules themselves. And that includes DNA methylation. Again, these modifications will help to determine whether the DNA is in the euchromatin state or the heterochromatin state. Main example we'll use here is histone acetylation. Enzymes are able to add this functional group, the acetyl group, onto histone molecules. And as a result of that, that tends to loosen chromatin structure, therefore promoting transcription. So here's an enzyme that can influence this histone molecule, which influences the folding of DNA, which ultimately can have an impact on whether those genes are transcribed or not. That's not a level of regulation that's available in prokaryotic cells. So here's our DNA double helix wrapped around some histone proteins. Here are histone proteins with these little tails sticking out from them. Okay? If those histones are unacetylated, then the nucleosomes tend to wrap up in kind of a tight ball, and this would be more compact and therefore not as readily transcribed. But if we acetylate the histones, that tends to promote chromatin loosening, and this ball kind of unravels a little bit. And now more of the DNA is exposed to these transcription enzymes, and therefore this is more likely transcribed than this is. So basically an enzyme determines levels of folding of DNA, which determines how likely that is to be transcribed. This is a relatively new phenomena. It's been discovered well since we were in school. And this is a very interesting level, kind of a macro level, at which gene expression can be regulated. Okay, I can see glazed looks. Good time to stop. Again, this is not the kind of thing that you're going to wrap your head around in one day. So we will come back and kind of try to get some of these high points as we move through some of these ideas about how gene expression can be regulated. But it's going to take some work, and this is something you should be reading about and trying to battle through now. Okay? See you Monday.
Hey, Dr. Browder.
Yeah.
I have some questions about the lab that we did this prior week. Do you know if you would have time on Monday?
Should have, yeah. What time?
The only problem with Monday is I have chem lab until 4. I have that hour from 11 to 12, but I know that's typically when.
No, I'll be here.
Okay.
Yeah.
All right.
Would you send me an email and I'll put it on my schedule?
Absolutely. I can
Okay.
I can email Natalie if that would be easier.
Why don't you stop down and have her put it on my schedule now?
Okay. Thanks, Dr. Browder.
Sure. Sometimes I find myself wondering, is this too much for this class? You know, the regulation.
Well, the
of gene
operators, I think you've got to have the operons.
Yeah.
I'm not sure about the DNA, the histone modifications,
expression.
but it's got to come. We got to begin somewhere.
Yeah.
The methylation thing is something